let's do that. So another good warning, if you're not comfortable with recording, just to let you know that we will record the session so that others who weren't able to make it today can take a look at it. So like I said, some of you might have worked with OMERO before, but you might not know who we are coming from Glencoe Software. I should also mention that Mina, my colleague, is also on the call today, maybe paying attention to chat and things. If you don't feel comfortable unmuting and saying something out loud, feel free to post it in chat, and Mina is happy to interrupt me at any time, should feel comfortable interrupting me at any time if there's some good questions in chat that we need to stop and cover. So Glencoe Software is responsible for, we're the sole commercial partner of the open microscopy environment. And so some of you might have worked with the open microscopy or OME team before. They're responsible for supporting the open source OMERO and bioformats, things that you use in tools like Cell Profiler and Fiji, and your own OMERO installation at UNC. And Glencoe provides commercial licenses to OMERO, a product that we call OMERO Plus. You are working with open source OMERO at UNC, but one thing that has been added on to that installation is a product called Pathviewer, which is a particular viewer especially suited for large whole slide images. And so I think some of you are working with maybe Axioscan data or other similar whole slide imaging data, and Pathviewer is a viewer that is really well suited for that. And so I'll show you some examples of image data in Pathviewer today. What we also do is a lot of support and custom development for people who are working with both open source OMERO and OMERO Plus. And so, one example is this training session here today to take you through some of the basics of OMERO and how you might want to work with it with your image data. So that's who we are. Meet and I are both on the applications team with Glencoe, and so we get to work with a lot of fabulous end users and hear all the interesting science that they're performing with OMERO. And so, like I said, I'm going to take you through some of the basics of OMERO today, but please interrupt. We really want to make sure that this speaks to you and how you want to use OMERO. And so it doesn't really have to be a set agenda. We want to make sure that it makes sense for everyone. Okay. So what I'm going to share today is not your own UNC server, OMERO server. This is one of our demo servers. So just be aware of that. The data won't look exactly the same as what you see when you log in, but I will hopefully show some generic enough examples that you can apply it to your own workflows as well. So I'm logged in here as a generic import user. When you log in, you'll see your name up here in the top right. And if you so desire, you can even come in here and edit your user settings here. I'll speak in a moment about certain reasons, specific reasons why you might want to edit those there. But that's the first thing that you'll see to orient yourself to OMERO Web is your name in the top right-hand corner. And you'll also see in the top left-hand corner the group that you're a part of. And so this is kind of the first concept that I want to talk about in terms of OMERO is the idea that you log in as a particular user, and that user is a part of particular groups. You can be a part of multiple groups, and there can be multiple groups within OMERO that you are not a part of as well. And so here, this import user is a member of two groups, this demo group and RW group. These two groups you'll notice are also different colors. So groups in OMERO are where we set the permission levels, okay? So for example, this demo group, I'm the only user, but here in the read-write group, we have two users. We have an import user and a test user. And so then the question becomes, if 2 people or 10 people are a part of the same group, so all members of the same group, what are they allowed to do within that group with each other's data, okay? And so that's what I mean by the permissions level. So you might be used to this on a file system, for example, whether you're able to read someone else's data or delete someone else's data. And the same concepts apply in OMERO, where the green group is the most permissive. It's a read-write group. That's what the RW stands for there. I purposefully named it RW group, but of course you could name this something that was more interpretable to your use case, maybe a lab name or some kind of demo group. And so read-write group means that you can do anything. So all the members can see each other's data, and they can even delete each other's data. So it's fully permissive, and it works great for, say, a demo group where if someone does accidentally delete everything, it's not so hard to recreate. But it's not a super common configuration for typical lab groups or core groups. What's more common is a read annotate group, and that's what you see by the orange color. A read annotate group means that everyone can see each other's data and you can annotate each other's data. There's lots of different things that I mean by annotation, and we'll go through that today, but it's basically adding some more information on top of the imaging data. And so you're able to add, but you can't subtract, basically. So you can't overwrite or delete someone else's work in a read annotate group. And then there are also more restrictive groups. So there's, for example, read-only groups where you can see each other's data, but you can't do anything to it. And then there are even private groups where you can only see your own data. So certainly there are a lot of different conditions where those different permission levels might apply. And so I'm happy to think about specific examples for your particular use cases, but read annotate is the most common configuration. It does get more complicated than just that sort of baseline permission level. I'm not going to go too deep into the weeds there, but you'll see that I'm in this category of members here. You can also have the role of a group owner, which this group currently doesn't have a group owner. That might be, say, a PI or a data manager that is a group owner, and that group owner has a little bit higher permissions even within the permission level of the group. For example, they can add people and take people away from the group. And so you can already see how that could certainly influence the overall structure of the group if you have the ability to add and subtract users. And then outside of the group context entirely, you have admin users. And so this is very high level admin. This allows you to create groups, delete groups, all of those kinds of very diverse workflows to manage the kind of structure of your OMERO installation. And so just want to point those two things out, that there are admin users and there are group owners that are sort of have greater permissions than just what's set at that group level permission. And so those might be relevant to some people on the call today, especially if you're sort of a lab manager or a PI, or you have a particular use case where you want to be able to create and do that sort of administration tasks. So happy to talk about that in, say, an admin training session with a smaller group, but I'm not going to get too into the weeds today, but please let me know if you have questions. Okay. So that's very high level when you log in, the fact that you are in. So I didn't really point that out here. So here I am currently in demo group. I'm a member of 2, but I see demo group here because that's the group that I'm currently sitting in. And I'm also viewing this demo group. I see my username here, import user. You'll see probably your real name there, but this user is first name import, last name user. And so I'm seeing this demo group in the context of what's owned by import user. Okay, so sometimes if you log in and you're sitting next to a colleague and you both log in and you see different things on your screen, even though you're both in the same group, a couple of things to check. Are you in the same group? And are you viewing the data of all members? Or are you viewing the data of a single member? Okay, so I can hop into this read write group, for example, and I can take a look at the data owned by test user. And I in fact think there's no data owned by test user. So it looks as if this group has no data in it, but I could instead go into all members. And I see that in fact, there is this data set here. And so this left-hand panel is going to change based on the context of the user you're logged in as, what do you have permission to see, and what group have you selected, and what user's context have you selected in order to see that data. Okay. So that's probably the first pass thing to do is, be able to log into OMERO and be able to see the data that you want to see. Okay, so I'm going to go back to demo group. I'm going to look at the data from all members. Okay, so I see this left-hand panel populated with some icons. And so I next want to go into what these different kinds of OMERO objects are. So we know that we have images in OMERO, but there are lots of different kinds of objects in OMERO that are used to organize that image data. And so I want to step through some of the language of OMERO that you might see. So the first is that you see these grayish blue icons, and those are called projects. And within that project, you see these green folders, which are data sets. And then within that data set, I can expand this, and here I see 4 images, okay? And so images in OMERO are 5-dimensional objects. You have X, Y, Z, and then you can have time and channel as well, okay? And so it would apply for your whole slide imaging data, your confocal acquisitions, your light sheet acquisitions within those 5 dimensions. Hopefully we have that covered. And each of these images is owned by a particular user. So when I refer to your data or my data and these permission concepts, I'm referring to the fact that each of these objects in Omero, including an image, is owned by a particular user. Okay, so this image is owned by import user, and the image also has a unique ID. And you'll find that every object within Omero has a unique ID. And so here, because this is an image, we have an image ID. Okay. As I was clicking through these projects and data sets, if you happen to be looking at the right-hand panel, you would see that, in fact, those objects also have unique IDs and owners. And so this data set is also owned by import user. It has a data set ID, and it has other metadata that you see populating this right-hand panel. And so, for example, I have data set details. I could add a description if I wanted, and it has a creation date. When was that data set created? Whereas if I click on the image, I see that there's a little bit different metadata here. So I can add a description here as well, but I also have things like the dimensions in X and Y, the pixel size, the number of Z sections or time points. And so this metadata is metadata that's populated upon import, and it's metadata that's parsed from that original file. Okay, so a lot of you are probably doing image acquisitions. And so you're probably very familiar with the idea that you acquire an image and there is pixel data within that file, but there's also a lot of metadata that tells you about that image. And so some of that ends up here in this section. Of course, there's a lot more metadata that's in this original file, and you can find that under the acquisition tab. And every file format is going to be different in terms of what keys and values are in that metadata from the original acquisition. But we try and parse out the really key bits of information and put it here. I know that we were talking with Michelle earlier that in some cases you might find that some of this information is missing. So maybe your file didn't have the metadata on the pixel size. And so you might see that this information is not populated. So that could be an issue with the file. Maybe that metadata was missing. It could be an issue on our end. Maybe that information wasn't parsed correctly. And so if you ever see issues like that, happy to get some feedback on where that information is missing, what the workflow is, and how we can help you repopulate that information. A lot of this information can be added post-import if you find that it's missing. Okay. So there is this hierarchy here of project dataset image. I'm going to stick within that hierarchy for now. There's another one that I want to describe in just a moment, but I'm going to stick with project dataset image for right now. That's really applicable to loose images. So whole slide images, confocal images, where you really consider sort of the single image, the entity of the acquisition. Okay. This is in contrast to plate acquisitions, which some of you might be doing. And so I'll get to those in just a moment. But this project dataset hierarchy really makes sense for those kind of collections of images. And so I spoke about the metadata in this right-hand panel that was populated from the original file, but probably, as we all know, there's a lot more biological or experimental metadata that isn't in that original file. Maybe that's written down in your lab notebook. Maybe it's in some piece of paper towel that you wrote it down on. It could be in a lot of different places. And so there's lots of ways that this can get into Omero, but I wanted to show you where it could exist in Omero, and certainly open to questions on how you might populate these different metadata fields within Omero. So and as I'm going through this, by the way, this is one example of an annotation, which I mentioned earlier. So I was talking about a read annotate group where you could read someone else's data and also annotate it. Some of the things I'll describe here are what would fall under the category of annotations. Okay. So there's two kinds of metadata that I want to highlight to start with that are relevant for putting experimental or biologically relevant metadata into Omero. And so these are tags and key value pairs. And actually, I'm sorry, I'm going to switch to a different data set here that has a little bit richer metadata. So these images are all from the Atlas of Intestinal Transport, which is a public data set from Jerry Turner's lab out of Harvard Medical School. So I just wanted to credit them for all the beautiful acquisitions that you'll see here and the metadata data that they've collected on this. So if you're interested, you can, if you Google Atlas of Intestinal Transport, you'll find this beautiful public data set. So that's what I'll go through to show you examples of this metadata. So I'm going to expand the tags section as well as the key value pairs section, because some metadata is duplicated in both of these categories. And so I want to highlight these two different kinds of metadata and why you might use one versus the other. So I'll start with key value pairs. This is like a dictionary. So on the left-hand side, you see all the keys, for example, diagnosis. And then on the right-hand side, you see the values, in this case, normal, okay? And so a set of key value pairs, or this is known as a map annotation in OMERO. This can be a really helpful representation of information because you have both the key and the value referenced within the UI of OMERO Web. And so I really like this. M might be unclear what that's referring to if you just saw it by itself, but okay, I can see that it's referring in this case to this patient's gender from which the sample was collected. Same with 4. What is 4 referring to? Well, it could be a number describing anything. In this case, okay, I know it's referring to the age and the units of that age in years for this particular sample. And so a great way to populate a dictionary's worth of information on the sort of conditions of the experiment or the particular sample that you collected, okay? Now, some of this information is duplicated here in the tags tab, okay? And so you see, for example, normal. You also see a patient ID and a couple of other tags as well. And so these, if you sort of already see compared to those key value pairs, I just see normal. I don't see diagnosis normal. I just see the value basically. And so why might you use a tag instead of a key value pair? It seems to be kind of information poor relative to the key value pair. So one reason why people like these is that, first of all, these are clickable buttons in the UI. So I'm going to click on this patient ID 31 here. And now I have switched into the tags tab on the left. So I was previously on the explore tab. That's where I saw all of my projects and data sets. And now I'm within the patient ID 31 tag. And you can see that it is a part of a tag set called patient ID. And so this is a new hierarchy that is completely separate from that project data set hierarchy. And so it's a way that you can organize your images in a lot of different ways. And so this image, for example, is a part of the patient ID 31 hierarchy, but it was also a part of, if I click this icon here in the right-hand panel, I can see what projects and data sets it's part of. And we just saw that just a moment ago, but I can see, okay, it's a part of the Atlas of Intestinal Transport data set. And so it's in these two separate hierarchies, but it's of course the same individual image with the same ID and representing the same file behind the scenes. Now, this image had some other tags as well. And so I can even click on the normal tag, and it's going to take me into this normal tag within the diagnosis tag set. And I see now a different collection of images. So again, a completely parallel hierarchy where the same image is in each of these different nested hierarchies without having to have sort of 5 folders deep worth of information. And so a single image can be represented in a number of different hierarchies that make sense for a particular experiment. Okay. So this tag set tag organization can be really helpful and is something that is not possible with those key value pairs or map annotations alone. And so you can strategically use tag sets and tags and key value pairs depending on how you want to organize your data and be able to explore it within OMERO Web. Okay, any questions on those two kinds of metadata and how you might use them? Oh, okay, great. So I'm going to then hop back to the Explore tab. And I'm going to talk about, so I talked about project data set, all of those whole slide images from the Atlas of Intestinal Transport were within the project data set hierarchy, and they had some tags and key value pairs used to annotate them. But now I'm going to talk about a separate hierarchy within this Explore tab, which is the screen plate hierarchy. And so I mentioned earlier that the project dataset hierarchy was really good for like loose images. If you have a collection of whole slide images or a collection of confocal images where the individual image is the entity, but some of you might be acquiring plates worth of data. And of course that is a collection of images, but it really matters the layout of that plate and what wells are where. And instead it might not be metadata that's at the image level, but it might be metadata that's at the well level. So for example, if you added a compound to a particular well, that might be metadata that's relevant not for an individual image, but instead for the well and all the images that were collected within that well. And so there is this separate hierarchy for screens and plates. Here I have one plate within this screen, but you could acquire multiple plates for a given screen. And so you'd have multiple entries listed here. And you can see in the center panel here that I see a little plate layout. And this is flexible depending on the number of wells in your plate. But here, this is a 96 well plate. And so I have my columns and rows, and I can click on one of these images, and I see that in this particular well, I actually only acquired one image. So if you're doing a plate acquisition where you take, say, 4 fields, 4 images within a given well, then you would see 4 little icons here in this lower panel, okay? So if I click on this image, I have the same metadata that I just highlighted on those whole slide images. So the dimensions are smaller, but the same kind of metadata is there. But in this case, I actually didn't populate any key value pairs at the field level or the image level. But instead, if I click on the well, you can see now I am looking at well B3. The well ID is 111 rather than the image ID. And here I do have some key value pairs that tell me what the compound and dosage was for that particular well. And so one really important concept in terms of tags and key value pairs is that they can be linked at a variety of points within these hierarchies. And so I highlighted image level key value pairs and image level tags, but here this is a well level set of key value pairs or map annotations. You could even have tags that are at the project level or at the data set level or screen or plate level. So any of these objects that I'm talking about, you can annotate them accordingly by adding a key value pair or by adding a tag, okay? And so in some cases, the entire data set has some experimental parameter that you want to link to the project level and not to each of those individual images, okay? Great. So separate hierarchy, screens and plates. I want to talk about next probably how we can get to the viewers from here. If there's no questions, I want to give everyone the chance if there's any questions on this metadata. I don't want to go too fast through it. Is everything clear in terms of what metadata you can add and how you can organize data?
Erin, I think I have I understand how it's organized, I guess.
Yeah.
And let me know if you're going to cover this later, but like, how do you get it in there?
Yeah, great question. So you can do it manually. I know that's not the preferred route. So you can, for example, I could start typing here, adding a key and adding a value, no matter where I am. If I click on the image instead, I can start adding keys and adding values here. And so in some cases, you've acquired 10 confocal images and you actually do want to sit and type and add those different values there. But everything that you can do within OMERO Web here is accessible programmatically. And so for those of you who want to import metadata or analytical results in a really custom way, everything is accessible through the APIs. And so you can use MATLAB, you can use Python, Java in order to put data into OMERO and read data out of OMERO. And some of us, want to be able to have that flexibility, but don't necessarily want to write the script ourselves. And so you can create custom import scripts for metadata to get metadata into OMERO. And there are also existing ones. So there are existing scripts that let you take, say, a CSV, an Excel sheet, and have a column with all of your image names, and then columns with whatever metadata you want. And so that column name is going to become the key, and then the value that's in each of those rows is going to become the value for that key within OMERO. And so if you have 10,000 images and you have a CSV with those 10,000 rows in it, then all of that data will be imported by that script. And fundamentally, that is using the API in order to do that. And so if you wanted to do that in a very custom way, you can do that. Or we have some scripts that I think cover the basis of a lot of the sort of standard import workflows for metadata, where you would populate those key value pairs and then even maybe turn some of those key value pairs into tags, for example. So that's how we actually populated this metadata, is we had a CSV with all of these key value pairs, we imported it, and then we ran a second step that said, okay, certain key value pairs I want to turn into tag sets and tags. And so, for example, diagnosis normal, diagnosis was turned into a tag set and normal was turned into a tag. And that's why I have the normal tag here with the tag set of diagnosis.
Cool, thanks.
Yeah, of course. Someone else was unmuting. Was there another question?
It was me. It was kind of similar to what Wendy asked, is how can you tell the software actually what wells is the image coming from? Because I mean, I usually, I don't know, image 10 384 well plates, so I wouldn't like manually do that. And I, like when I use Cell Profiler, for example, it is easy to tell him how to get that information from the name of the image, because it has the well.
Mm-hmm.
And so I was wondering if there was something similar also here, or you had to actually write a code for it. Or maybe it's available somewhere and I can just use that code.
Yeah, no, another great question. So it depends on the format of the data. So a lot of plate-based formats already have all of that metadata within, say, the individual files or within a companion metadata file. And so OMERO is using bioformats, which has readers for all these diverse file formats that we use in bioimaging. And so depending on the format, you could import them and it will just work. So OMERO bioformats will interpret that metadata correctly, parse that metadata correctly, and you'll get the right layout of the plate within OMERO. The other possibility, and this was actually the case for this plate, so I should also note that this is from the Broad benchmark collection. So you can find these in some of the Cell Profiler documentation, and I think it was even used in some of these cool segmentation challenges, like those that help develop Stardust, for example. And so this is also public data, and this data is a big folder full of TIFFs. And so we first imported it here as 96 TIFFs. And then we took all of these images, and there's, again, one of these standard scripts that comes with OMERO. I'm not going to dive too deep into OMERO scripts in this training, but we can certainly talk more about them in the next training, where we'll talk all about the OMERO Cell Profiler connector that you all are using and how you can run Cell Profiler image analysis from OMERO, from the browser, or in the desktop GUI with images in OMERO. So we'll talk way more about that, and you'll see some examples of OMERO scripts. But here, there are some standard OMERO scripts that get shipped with every OMERO server installation. And so you'll see that you should have an OMERO folder here with a number of different import scripts. And there's a dataset to plate import script that can take a dataset full of TIFFs and turn it into a plate. And that's exactly what we did here. And so happy to demo that in that Cell Profiler session, or maybe we should have a more in-depth like API and scripts workshop for those who are so inclined. But that's exactly how you could do this, where based on the name alone, you use that to populate a plate. But for some formats, it should just work. You import the plate, for example, a CZI plate from the Cell Discoverer 7, or even some of these like MetaExpress, these really true sort of high content screening systems, the plates just import based on the file metadata. So happy to take a closer look at some of your plate acquisition examples.
Okay, thank you.
Of course. Okay, great. So yeah, lots of different examples of how you can get both image data and its metadata into OMERO. So great questions there. I want to sort of transition to talking about the viewers because we do have Pathviewer installed at UNC. And this is actually sort of a newer addition. So even some of you who might have been using OMERO for a number of months, maybe you haven't yet seen Pathviewer. And so I want to start with just maybe a single image. So I'll go into my Atlas of Intestinal Transport and I can just open up one of these images. I just double clicked it and it will open up in Pathviewer. Okay. You can also go to the right-hand panel right next to full viewer. You'll see the open with menu. And if you have multiple viewers installed, you can choose which one you want to use. So maybe you're using iViewer as well. And so you can select specifically what viewer you want. There's a number of open source viewers and viewers supported by Glencoe Software. So we certainly see a lot of installs where you're choosing what viewer you're using for a particular use cases. Okay. So I just double clicked on the thumbnail there and it launched Pathviewer. And here again, these are images from Jerry Turner's lab from the Atlas of Intestinal Transport. And here, this is a 4-channel image. So just to orient you to your view here, so this is Pathviewer. This is the viewer that I mentioned earlier that is really well suited for these whole slide images. In the left-hand panel, I have by default, if I double click on a single thumbnail there in my dataset view, Pathviewer will open up that image that I double clicked, but you'll also see in the left-hand panel all the thumbnails for the other images within that dataset. And so that's what you should expect is populating this left-hand panel here. And then in the center, you have the image itself. And then on the right-hand side, you have some of that core image metadata that we saw in OMERO Web, in addition to any description or key value pairs that you have on that particular image here. So I see those here. And then I also see a bird's eye view. So as I zoom in on this image, I can still see the context of where I am in the larger whole slide image. You know, as some of you who are acquiring these whole slide images might know, you could easily have, 50K by 50K pixels or more. And so you might be zooming in on some very small structure and you sort of forget where you are in the broader context. And so that bird's eye view can be helpful for that. In this top panel, you'll see your channels themselves. And so you can turn off particular channels, you can change the color of particular channels, and you can adjust the range that you're viewing from that particular channel. For those of you who are working with highly multiplex data sets, so maybe you have more like 20 or 30 channels instead of 4, you might hop into the viewing options tab instead, where you have a little bit more screen real estate dedicated to your channels. And so here I can expand and scroll through each of my individual channels, again, adjust the contrast, turn channels on and off, etc. And you can save these rendering settings for the future. So if you spend some time adjusting these rendering settings, you can save them so the next time you come back to the image, it looks the same. Okay. The other concept here is channel groups. This again is really applicable for those of you who are working with highly multiplexed data sets. This image here has just 4 channels, and so it's a bit overkill to create groupings of channels, but certainly you might want to create particular visualizations based on certain channels being on and off. And so I can sit and say, oh, okay, I want DAPI and M-Cherry. So let me turn these 2 channels off. And now I'm taking a look at DAPI and M-Cherry. Well, now I want to see GFP and Sci5, so let me turn off these 2 channels and let me turn these 2 on. And okay, that's not too many clicks to turn those channels on and off, but maybe there are particular visualizations with particular channels turned on and off that you want to view over and over again. And so it might be interesting to create what are called channel groups, or particular collections of channels. And especially if you start having 10, 15, 20 channels, this can be a really nice workflow. And so I can create a channel group. I can name it whatever I want, and then I can click and drag channels into that group. And so maybe one that I want to see is DAPI and M-Cherry. And another group, I'm actually going to copy DAPI to the other one. So I, in this case, just held down my command key, which allows me to switch between copying and moving channels between groups. And so maybe I also want to see DAPI and Sci5. And those are, this will be group 2. Oops. Sorry, group 2. Okay, and we'll save those groupings. And so now I can just turn, and we'll turn off things that are ungrouped. Now I can switch between these two groups with fewer clicks. So I'm going to turn off group 2 entirely, and I'm going to turn on group 1, off group 1 and on group 2 entirely. So you can switch between them with slightly fewer clicks, especially when you have high numbers of channels. And so that's the channel grouping context. Oh, I saw Shannon, did you have a question? Yeah, go
Yes. So if you set this up in one image, could you go to another image and use the same groups without
Yes,
recreating?
exactly.
Okay.
Yes, absolutely. So a couple of options there. You can copy the channel groups, and then you could go to another image and paste them, or you could select multiple images here and paste to the selected slides. So that's one option that is still slightly manual. The other option is that there are command line tools to generate channel groups. And so if you did have like a really massive acquisition with tens of thousands of slides, for example, and you wanted to, or even hundreds of slides, and you wanted to apply all of those channel groups in more of an automated fashion, you could even do it via those command line tools so that you could, upon import, immediately create the right channel groups across an entire data set of images, for example.
Great, thanks.
Of course. So I'm going to come back to these channel groups in just a moment when I talk about Pathviewer Grid, which allows you to view multiple images at once. So I'm going to skip some of these icons here that you see in this window here, but we'll come back to it in just a moment. But hopefully this just illustrates the idea that you can create collections of channels and create interesting visualizations with those channels. The other element that I wanted to highlight while we're in core Pathviewer is this center panel here where I have some navigation controls and some annotation controls. Okay, so I can zoom in and out on my image with these controls here. I can rotate my image here. But I can also annotate my image. And that's one of the other core use cases for Pathviewer is that you want to annotate your whole slide image. And this gets us back into this sort of annotation word. Okay, we talked about tags and key value pairs. Regions of interest or shape annotations are another type of annotation within OMERO. And so I might want to draw a particular style of ROI. So here, let me turn off my bird's eye view for a moment. So I've selected the freehand polygon tool, okay? And you can see that it's giving me the option to change my default ROI style even before I start drawing. And so I might want to change the stroke color or the fill color. I can make either of these more or less transparent. I can choose a dashed stroke if I want to. If I am going to give this some label, then I can change the font color of that label, okay, and the font size. And then, okay, I can finally start drawing. Now you could change all these features after you draw the shape. And okay, we see it here, and I see my label right in the center, and I see, let me just hide this a little bit so we can get a little bit more screen real estate. You can see my ROI in this center panel here, the text label in red, and you see my green dotted line outline based on the selections that I made here, okay? I also can set a name that is different from the label. In most cases, we just duplicate the label to also be the ROI name, and you'll see this in this right-hand panel here. So that allows you to navigate. If you've made multiple ROIs, you can navigate that list of annotations, okay? There are many different kinds of annotations. So there are rectangles, ellipses, etc. But what I also wanted to highlight here is that this ROI itself can include a description and it can include key value pairs. And so just like we saw at the image level or the well level or the data set level where we could add those tags and key value pairs, here in Pathviewer, we can even do it at the ROI or the shape level, okay? So here, this particular polygon, maybe I want to say this is class A, and is it normal? I might want to check true or false here. And so lots of different ways to annotate a particular ROI. And importantly, I might create a separate ROI. So let's create one here, and this might have a different set of key value pairs. And so if I click between them, I'll click this one here. This one has the key value pairs that I just created. This one does not. And so those annotations, those key value pairs are annotations on the region of interest annotations. And so they're not at the image level, they're one level deeper. And so you might highlight different structures and have yourself or your colleagues comment on those structures. And so yet another level in this hierarchy of where you can provide that annotation level information. Okay. Any questions there on slide annotations and options? Okay, so I have another example of doing annotations where potentially you have multiple images that you want to annotate at the same time. And so this brings us into another sort of parallel product of Pathviewer, which is called Pathviewer Grid. Okay, so I'm going to go back to the web client, and I'm going to go to my patient ID that I had earlier, patient ID 31. You might have noticed this earlier, that clearly these tissue sections are looking very similar, right? So they're serial sections from the same patient. And so what I might want to do is I might want to view each of these images side by side and try and annotate the same structures, okay? And so I could open up any single one of these images in Pathviewer, but I could instead select multiple of these images. And then I could open with Pathviewer Grid, okay? And this is just like having a single Pathviewer Grid, but now I have multiple, so 2 side by side. Right now my grid orientation is 1 by 2, but I could increase this. And so, you know, maybe you have 20 images that you want to view side by side. That's totally possible here. And importantly, I can synchronize these views, and then I can navigate them at the same time to try and, you know, find those same structures and annotate them accordingly. Okay, so you can see these are not the same section; they are serial sections, and so some of the structures are slightly different. And so one thing that I might want to do is I could unsync them. I can use a shared cursor between the two. So now I see these crosshairs on the 2 views. And then, okay, maybe I want to annotate this circular structure here. So I'm going to try and kind of move it around and get my crosshairs centered in both images there. So I'm moving one of them in X and Y relative to the other. And then I'm going to synchronize these. And now I can, you know, zoom in and out in parallel fashion between the 2 again. Okay. And then I'll come in here and I will annotate on one of my images, a nice freehand polygon. And I will just copy this, and instead of pasting it here within this window, I'm going to paste it to all of the views within Pathviewer Grid. And so now it shows up on this other image. And so of course, if you had 20 images open at once, it would paste to all of those different views. Okay. And then you don't have to go through and save each individual panel, although you can if you want to. You could instead save across all of your views. Okay. And so Pathviewer Grid might be quite useful if you have serial sections or some other sort of matched acquisitions where you want to view multiple images side by side, annotate them. And of course, you have the same ROIs tab here where you could add descriptions and key value pairs on that ROI. And so you can do all the same annotation workflows within Pathviewer Grid with multiple images side by side. Okay, great. So I mentioned that there was another workflow that I wanted to highlight with channel groups. So here, now that you've seen Pathviewer Grid, this was the kind of original concept for Pathviewer Grid, is that you might have separate images that you wanted to view side by side, right? But for those of you who are acquiring these multiplex data sets, you're well aware that a single image can be quite a lot of information in its own right. You don't necessarily need to have 2 images that you're looking at once. If you have 30 channels, that's a lot of information to digest, and you kind of need more than one view to be able to capture that. So in Pathviewer, you can, when we are looking at our viewing options tab, here I have 2 channel groups, remember, that I created, one with DAPI and mCherry and one with DAPI and Sci5. And I can use these icons here to open this image up in Pathviewer Grid, okay, and just this image alone. And so either I can view each of the individual channels in grayscale, or I can view the individual channel groups side by side in Grid. So I'm going to use that option here. I think it's telling me I have unsaved changes, but I think I have saved my channel groups. Okay, so here, this is my single image now. So it's actually the same image. If we look at the titles here, it is in fact the same OME TIFF. But now I have it split with my DAPI and mCherry channels and my DAPI and Sci5 channels. And so now I can synchronize these views. I know I don't have to do any shifting in X and Y because they're the same image, so they're already aligned. And I can navigate them and explore that Sci5 signal and that mCherry signal side by side one another. And so this can be really helpful. Like I said, if you have a large number of channels, you probably can't turn on 30 channels at once and appreciate all of those colors. And in fact, Pathviewer won't let you. There is a configurable maximum of how many channels you can have turned on at once. And so you could instead create groupings of channels or view individual channels side by side in grayscale and navigate them simultaneously. So you have two parallel views. Remember, you have your shared crosshairs, and so you can take a look at the Sci5 signal and see if there's comparable mCherry signal in that particular cell. This was in part inspired by, we saw some views in QPath, for example, where you can view side-by-side grayscale views, and that was really popular with some QPath users. So I don't know if anyone there working with whole site images is working with QPath, but maybe you've seen a similar kind of workflow where you can view individual grayscale channels side by side. And that's the intention here. You have a lot of channels. How can you explore that data, the richness of that data side by side? Okay. Any questions on those sort of channel generated Pathviewer grid views? Great. Okay, cool. So I think at this point, I will sort of transition to talking about the plan for next time, just so we have everything covered. I do have one more thing that I can show on exploring metadata as kind of a preview for next time, but I want to make sure that we don't run up against time because I see that we're approaching the end of the hour. So just wanted to mention, if anyone has to scoot out right at the end, we are planning on having another session. I think it's going to be in approximately 3 weeks. And in that session, we're going to talk about the OMERO Cell Profiler connector, where you can run cell profiler analysis via the browser, or you can use the traditional cell profiler desktop GUI and use it with images that are sitting in OMERO. And so that will be a little bit of a deeper dive into how you can perform that cell profiler analysis and how you use things like OMERO scripts, which I mentioned earlier. And so I'll go through sort of what OMERO scripts are and how you interact with them in a little bit more detail. It'll be in the context of the OMERO Cell Profiler connector, but I'll highlight some of the others that I mentioned here today in terms of importing metadata so that you can see those examples as well. And then, of course, one big component of the connector to Cell Profiler is that you'd want to be able to then bring in the results from Cell Profiler and explore them in OMERO. So that's what I kind of want to preview really quickly. So I'm not going to show you how the Cell Profiler results were generated, but I'll show you once that data is within OMERO, how you might kind of mine through that a little bit. So on this particular screen, I have what's called an OMERO table. This is effectively just a table. You can think of it as a CSV. It's not actually stored as a CSV, but that doesn't really matter. If I click this eye icon here, it's a table that I can explore in the browser. I see all of my column names here. I see my plate ID and the well ID, and I see, each row has these particular measurements. And these are measurements that were performed in Cell Profiler. And so you'll see how we acquire those next week. Okay. Sorry, not next week, next session, which will be in about 3 weeks. And so how might I explore these results and mine through my plate data within OMERO? And so one plugin that is open source is called OMERO Parade. And so when I previously clicked on this plate, you saw the plate layout here and all of these thumbnails, but I'm going to switch now into viewing Parade instead of the thumbnails. And at first glance, it's not going to look any different. We still see all of our thumbnails here. By the way, if you had multiple plates, you could kind of scroll through them here where you had plate after plate after plate and all of the thumbnails. But you also see these new fields up here where you can add filters or tabular data, okay? And so One thing that I might want to do is I might want to filter based on some metadata. And so maybe I have a key value pair. I know, for example, that this plate had particular compounds applied to it. And so I'm going to select one of those drugs, and you see that the wells were filtered out. And so now I just have this subset here where the particular compound was used in these wells, but not the others. And so the others are kind of grayed out. Okay. I also might want to filter based on my tabular data that I just showed. And so, for example, I counted the number of nuclei in each of my wells, and I might want to filter out the wells that have a smaller number of nuclei. Maybe that suggests that the acquisition wasn't so good, the cells weren't as happy, and so I want to filter any wells that were below that particular threshold. Okay. And now I want to start to explore this data. So, okay, these are my candidate wells that I want to take a closer look at. And so I can maybe hop into my tabular view. So right now to start, I just have my thumbnails and the names of those images. And I'm going to start adding in some of my tabular data. And so I know in this particular case, I know that this was a translocation assay. And so we're looking for some of our yellow signal to be moving into the nucleus. And so I can already sort of tell which columns might be kind of interesting here, but you might be doing this in a less informed way, first time exploring your data, but I'm going to kind of cut to the chase more quickly. And so I will go down to my, for example, my cells and look at their form factor. So what is the shape, that sort of morphometric feature of my cells? And then I'm also going to look at the intensity of the mean intensity of the GFP signal. And so I have cells, intensity, mean intensity, GFP. For those of you who use Cell Profiler, these kind of columns and there's the naming might be familiar to you. So, okay, now I have a simplified version of my table with the columns that were of interest to me. I can do a couple of things. I can color code these values, so I get a little bit of a heat map. I can do that for both. And then I can even sort by one of these columns. And so let's sort by form factor. And I can kind of scroll through these and get a sense of, oh, okay, these are looking like they might be somewhat correlated, which of course I selected them because I already knew that might be the case. And okay, I can even hop into a fairly simple 2D plot. Sorry, I have my view zoomed in a little bit, and take a look at how these two variables relate to each other. So the form factor and the mean intensity. And so I can see that I kind of have clusters of two groups of cells. And I might want to say, okay, these cells here, what might they have in common? And sorry, these are images rather than cells. Of course, there's many cells that were averaged within that single measurement. So I've selected this cluster here. I can now expand key value pairs, for example, and take a look at all of the key value pairs for each of those wells. So well A8, A9, A10, et cetera. Those are all the wells that I have selected here, okay? And I see the drug that was used. That's not surprising because we actually filtered the wells based on the compound that was used. But I also see the dosage, okay, 7, 8, 9, 6, okay. And now I can come in and select a different group of objects, of wells, and I look at the dosage and okay, it's a bit lower. And so maybe there's a dosage dependence to this sort of difference, these two clusters of data. And so, well, maybe now I want to take a look at these images. And so a couple of ways you might do this. One way that I like is, I might again highlight these objects here. And then if I hop back to my plate view, those images are now highlighted. And so you see them just outlined here. And I sometimes find that a helpful view. I can zoom in these thumbnails here. And what you might be able to tell from this view, I know it's over a screen share, is that these objects are looking a little bit more compact than the cells in this image here. And so I can even click on one of these objects. So let's get this one here. I can hop over to the preview tab and say, okay, it looks like a lot of that yellow signal is within the nucleus. And I can look over at these images and, okay, that yellow signal is clearly not translocated to the nucleus. So in your translocation assay, you can clearly see some of these groups starting to form. And okay, I came in with a fairly informed perspective on what I was looking for here. And so you might go through a much longer list of iterations in order to get to this point. But that's how you could sort of start to explore or mine through your data from something like Cell Profiler or from some other source. So this is OMERO table that I showed that has all of these well level results could be generated by Salt Profiler, but it could be generated by your own custom Python script. The last thing that I'll say on OMERO Parade is that I showed it in the context of a plate, but it's also completely applicable at the level of a project data set. And so if you, for example, have the Atlas of Intestinal Transport, you can add the appropriate filters. So maybe I want to filter by a particular tag. So let's look at the images. I think they're all actually diagnosed as normal. And so we can select a particular subset based on a tag that was on those images, and then see the thumbnails accordingly here. So it also applies to that project data set hierarchy. And here I could expand multiple data sets. Let's remove this tag, and I could view, navigate through multiple, let's do TCJ, ACC. Now I can scan through multiple data sets at once within OMERO Parade, just like you could multiple plates. So both hierarchies apply to OMERO Parade. We just have a couple minutes left. I know that it was mentioned that there is some interest in 3D data. And so we do have some examples loaded into this demo server here. I wanted to emphasize that OMERO in general, a lot of the viewers are plane-based viewers. And so if I did want to open up this particular data set, which has a Z component to it, I can open it up in Pathviewer. And I do have this slider that allows me to select through my Z planes. But I don't have a way to render a 3D view, sort of a volume viewer of this object. There is a viewer called OMERO FP Bioimage. This is an open source plugin for OMERO using the FP Bioimage viewer that will give you a nice sort of volume rendering of this object. At Glencoe, we don't support it, not that we don't support you using it, but we're not sort of maintaining and developing that. But it's certainly something to look into. And the other thing you could explore is things like the OMERO Fiji connector, where there are, of course, a lot of interesting volume-based viewers. And so with that Fiji connector, you can open up images from OMERO in Fiji and then use whatever plugins you want. And so I just wanted to briefly mention that 3D data and time-based data is completely compatible with OMERO, and you'll find plane-based viewers with sliders. But if you really want that 3D rendering view, you'd want to sort of connect to some of these other applications that have proper volume viewers. It's not necessarily a focus of a lot of OMERO viewers, to be perfectly honest. Okay. So like I said, we'll have another session to dive into some OMERO scripts and specifically the OMERO Cell Profiler connector. Please let me know. I know there was some information gathering ahead of this session for particular interests and use cases that people had. So that was really helpful. So thanks for answering that survey, and thanks to Tessa and Christina for sending that out. And certainly keep giving us that feedback so that we can make sure these sessions are really useful and address any questions that weren't covered today. Okay. And happy to stick around for a couple more minutes if there are any questions. But thank you so much for coming and for the questions that you have asked so far.
Yeah, there were two questions in the chat there. In one, you kind of like answered the 3D images, but like NII files in particular, are they supported?
NII files can whose question was that? That was
Felix.
Felix. What's that instrument? Is Felix still here? Yes.
This is brain image.
Yes. Yeah, so these are like MRI scans that are obtained in Nifty files.
Okay. So let's take a closer look at it. I know that we have seen some MRI data in OMERO, some in the DICOM format, and that NII file that's not an extension that I'm familiar with, so we could give it a try. And let me just, if you have a moment, let me just take a look at our list, because it might be that it's supported and I'm just not familiar with it. Let's see. And the current OMERO deployment, I would sort of defer to Michelle, Christina, and Tessa on that question in terms of the details of the UNC deployment. Oh, and it looks like we do support the Nifty format. So apologies, Felix, that it's not something I'm familiar with, but it is on our list of supported formats. And so nominally that should be fine, but let us know if you have any issues.
Thank you.
Any other questions? Okay, great. Well, again, thanks so much for joining. I will send the link of the recording after this, so if anyone wants to rewatch it or send it to your colleagues who weren't able to come today. But thanks again so much for joining.